Okay, we're going to do this lab. You know, take the lab instructions, copy them, print it out, write them down, put it to the side so you can refer to them. Because we're going to do this Punnett square lab, okay? So we're going to go back to the lab link and open it up. Okay, the first thing is we watch this.
This Punnett square predicts the result of a cross between two heterozygous tall pea plants.
So this shows us how to do it.
Carries only one of the two alleles that controls height in
Okay,
pea
you've
plants.
watched that. Let's get rid of that. Save time. Okay, now we're going to do the experiment with it. Now it says, make a cross between the following flies. A heterozygous gray-bodied fly. Okay. Heterozygous means it's going to be a GG. Okay. Here's a gray fly. Okay, so GG is heterozygous, so we're going to select that one, okay? And a heterozygous gray-bodied fly. Okay, so we're going to do this one against one just like it. So we're going to pick another GG. So now we have GGs. Okay. So now, okay, we got to check the parents. Okay, so now it puts these squares for us. Okay. So now we're going to check the offspring. Well, if this is GG, what's the offspring going to look like? It's going to be gray, right? It's going to look like that. If this is G times small g, which is black, it's going to still be gray because it's dominant. So that's how we do that. G and small g, again, we put that one there. And then we put that one there because that's double recessive. That's going to be black. Check offspring.
Fill in the rest of the Punnett square to determine the expected genotypes and phenotypes of all the offspring.
Okay. So they want us to take this and put it there, and then this and put it there, and then this and put it there, and then this and put it there. So now we've got the whole Punnett square put in. So now we can take this table down here and we can fill it in. Okay, the scenario number 2, there it is, 2. Genotype of parent 1. Okay, G and little g. Genotype of parent 2. G and little g. Okay, so what is the ratio of the offspring genotype? Well, genotype is 1 and then 2 of those and 1 of those. So the ratio is 1 to 2 to 1. And then the next box we fill out is ratio of the offspring's phenotypes. Well, there's 3 grays and 1 black. So it's 3 to 1. Okay? So we can set this table out of our way, I think. I want to get it out of our way. Well, maybe it'll stay that way. So we got that one done. Now let's do another scenario. Return, reset. Okay, scenario 9. Make a cross between 2 flies that are expected to have all vestigial winged offspring. Ooh. So vestigial winged, well that's a wing. So this is vestigial wing. Okay. Oh, parent. Now how would we get all vestigial wing? Well, if vestigial wing is double recessive, then it's this way. Then we've got to have that one. And if all of the offspring are going to be double recessive, then we can't have any large L's in here at all, right? So we got to have one of these times one of those. Is that right? No, wait a minute. We got to find this one. Oh, that's what vestigial winged is. So it's a dominant gene. Okay. So let's take what parent 2 is. Now, we could do that. Let's see if it'll check parents.
Examine the parents described in the scenario.
No, they don't want us to do this one. They don't like what I did. Let's see.
Examine
No,
the parents described
I
in
got
the
the wrong
scenario.
parents.
Then try again.
Oh, maybe we want to have that one. Let's try it.
Examine the
No,
parents described in the scenario.
it's got to be this one
Then
then.
try again. Examine the
Oh,
parents described in the
we get it
scenario.
wrong.
Examine the parents described in the
Gosh,
scenario.
what am I doing
Then
wrong?
try again.
Well, if they're going to all have vestigial wings. Oh, this is the vestigial wing. Okay, I got the wrong picture. Okay. All right. I was right in the first place. Okay. I was right in the first place. I was looking at the pictures wrong. All right. So now, what are we going to have? We're going to have all of them are going to be like that. Okay. Check offspring.
Fill in the rest of
Yes.
the Punnett
Now
square
fill in
to
the rest.
determine the expected.
Well, the expected value here is a ratio of 1 because everybody is the same, right? So we go back to our table. Scenario number 9. Okay, I guess we need to print this table out or pull it off to the side so we can keep going with it. I got to get it out of the way. Anyway, so we can see everything, we got to get it out of the way. So it's scenario number 9, genotype of parent 1 is LL, well, yeah, and parent 2 is small l's, and ratio is 1. And the ratio, oops, gotta get over there. Ratio of the offspring phenotypes is 1. So if you have problems like I'm having, 1 means everything's exactly the same, which it is in this case. Okay, so we can reset it. You know, turn is what? No, that's there. So we're going back to this. And we got to get this out of the way. Anyway, I can't get that out of the way. So I would just write down the headings, set up a table in a Word document in another window, and then you can go on with doing a bunch of these. Okay? So, do monohybrid genetic crosses, which is we're looking at one trait at a time, and analyze the genotypes and phenotypes that are expected from each cross based on these scenarios, okay? So you go through a bunch of these scenarios. And then click the notebook to perform genetic crosses. We did that. Punnett squared, read the scenario at the top of the screen. Here's all the directions. I just showed you how to do it. So that's all there is to it. And you fill out the table and then figure out. But see, what I had problems trying to recognize what was what. But you move these things around. You choose what parents that you want. Choose the one you want. And then you can do a cross, see? Check parents. No. Make a cross between two flies that are expected to be 50% heterozygous long-winged offspring and 50% habitual. So you keep playing until you get it right. No, reset. Anyway, that's how you do it. Not much to it. Okay, I'm going to do done.